Good day, everyone. Hello, grade 10 students. Today, we will be having an in-depth lesson about protein synthesis. This is Teacher Divine saying learning is fun. In this particular lesson, you will gain knowledge about how proteins are made using information in the DNA. More so, at the end of this lesson, you will be able to differentiate DNA from RNA and explain how those genetic materials embedded in our DNA work to form proteins. But first, let us have a quick overview about DNA and RNA and its chemical composition.
There are two types of nucleic acids. First, we have the DNA, which stands for deoxyribonucleic acid. Is found within the nucleus of every cell. Your DNA is like your thumbprint. It is yours and yours alone. DNA holds the genetic information needed to make and control all cellular activities within a living organism. In 1953, James Watson and Francis Crick worked out that DNA is like a double helix. A helix is a screw-shaped spiral. The DNA is double helix because of the two strands which wind around each other. The double helix is like a winding staircase. The two sugar phosphate backbones make up the sides and the base pairs make up the rungs or steps of the winding staircase. Second is RNA. RNA stands for ribonucleic acid. Like DNA, RNA is a long strand made up of building blocks called nucleotides. But unlike DNA, RNA has a single chain and does not entwine in a double helix. RNA is a single-stranded molecule with no helix. The four bases are guanine, cytosine, adenine, and uracil instead of thymine. Let us take a closer look in nucleotides. A nucleotide is composed of phosphate group containing a phosphorus and oxygen atom that is attached to the sugar. So at the center of each nucleotide is a 5-carbon sugar, and then it is in deoxyribose form in DNA and ribose in RNA. Attached to the sugar is a nitrogen-containing base made up of 1 or 2 rings depending the sugar and the phosphate are the same in all nucleotides. Nitrogenous base will only vary because this nitrogenous base there are 5 types: adenine, guanine, cytosine, thymine, and uracil. Thymine is only found in DNA, while uracil is only found in RNA. This time, let us have a comparison between DNA and RNA. For sugar, in DNA it has deoxyribose, while RNA has ribose. In bases, DNA has adenine, thymine, guanine, and cytosine, while in RNA, it has adenine, uracil, guanine, and cytosine. In terms of strands, DNA is usually double-stranded, while RNA is single-stranded.
This time, you have learned the difference between DNA and RNA. Now, translating the code of life from DNA to RNA to protein. Proteins control the activities of the cell and so the life of the entire organism. But how does the DNA make a unique protein that will perform a special function? So this time, I will show you the steps in making proteins.
Protein synthesis is the process in which a cell makes a protein based on the message contained within its DNA. Steps in protein synthesis. In this given illustration, I will be elaborating the protein synthesis process. Transcription is the first step which happens inside the nucleus. The DNA is found inside the nucleus. Okay, this encircled one here. The genetic information from the strand of DNA is to be copied or transcribed into a strand of mRNA or messenger RNA. As you can see in here, the DNA unzips. This is also like a zipper. If you unlock, it will unzip. So enzymes split apart base pairs and unwind the DNA double helix. Take note that the DNA has a nitrogenous bases which are adenine paired with thymine and cytosine paired with guanine. We have these split pairs here. Adenine is paired with thymine and cytosine is paired with guanine. As the DNA unzips, the RNA is now ready to copy or transcribe the genetic information from the DNA. Bases pair up, so free nucleotides in the cell find their complementary bases along the new strand with the help of the RNA polymerase. So as the bases paired up, a new backbone forms. Sugar phosphate backbone is assembled to complete the RNA strand and separates from the DNA strand. This is now the new backbone form. Note that the thymine in the DNA strand will be replaced with uracil in the RNA. The copied information called codon, this one here is an example of a codon AUG, which is made up of 3 consecutive nitrogenous bases in the mRNA. This codon calls for an amino acid is sent to the cytoplasm where it will enter the ribosomal RNA, this one in here. Once inside the ribosome, which is located in the cytoplasm, translations begin, meaning the transfer of RNA or transfer RNA from the cytoplasm starts to bring the anticodon, also a 3 consecutive nitrogenous pairs that fits the messenger RNA code. Each anticodon represents one type of amino acid. UAC is an example of anticodon which represents an amino acid called methionine or MET. Why this happens? So mRNA has a codon of AUG, this portion here. The anticodon that will fit to this codon will be UAC because adenine must be paired with uracil since it is now an RNA. And uracil will be paired with adenine and guanine will be paired with cytosine. Each 3-letter combination is a code for an amino acid. Amino acids are the building blocks of protein molecules. When all codons are converted into amino acid, translation stops and a protein molecule is produced. This is now the new protein formed. This one is an example of linked amino acids which forms a protein. And they are linked together by a polypeptide chain. So hundreds of amino acids are linked together to make a protein. This is the other example of protein. As you can see, amino acids are linked together by a polypeptide chain forming protein. There are 20 types of amino acids. In the table given, it shows different kinds of amino acids and its abbreviation. For example, an amino acid alanine or ALA for 3-letter abbreviation. Another is amino acid arginine or ARG and asparagine or ASN and so on. So, those are the different steps in making proteins.
There you have it. You have learned the steps in making proteins. This time, let us have our activity for us to check if you have learned something from our lesson.
Now, let us start the activity. You will determine if the given statement is true or false. Number 1. Proteins control the activities of the cell. Is it true or false? The correct answer is true. Very good. Number 2. DNA can be found inside the nucleus. Is it true or false? The correct answer is true. Very good. Number 3. Codon is made up of 2 consecutive nitrogenous bases. Is it true or false? The answer is false. It is false because codon is made up of 3 consecutive nitrogenous bases. Next number is number 4. The job of RNA or ribonucleic acid is to carry messages from the DNA in the nucleus to the ribosomes in the cytoplasm. Is it true or false? The answer is true. And our last number is number 5. RNA is the building block of a protein. Is it true or false? The correct answer is false. RNA is not the building block of a protein. It's the amino acid. Congratulations! You get a perfect score.
That's all for today. I hope you enjoy learning. Till our next lesson. Bye!